adrenergic nervous system. They both intertwine, but in general, you have the parasympathetic nervous system, which is your rest and digest. And we'll see that on the slides. Rest and digest meaning, bless you, we are going to lower the heart rate. We're going to lower the blood pressure. We're going to decrease respiratory activity. And we're going to be increasing the blood flow to the stomach, meaning for digestion. So it's kind of like your depressant state. When we have the adrenergic system, which is modulated by the sympathetic nervous system, that is your fight or flight. So that's your adrenergic receptors. That's where you have, if they're activated, you have an increase in heart rate, blood pressure, your fight or flight, your skeletal muscles are active, there's blood flowing through you, you're ready to run, or you're ready to fight a tiger or whatever danger is nearby. So that's the kind of the two systems we're talking about here. And there's different pathways, but they're all, in terms of the nervous system signaling, it's similar between both the pathways, and there is an interplay with the neurotransmitters, meaning acetylcholine, right? Acetylcholine is the main neurotransmitter of the parasympathetic nervous system, but it's also involved in the sympathetic nervous system. It is the most prevalent neurotransmitter in the body. If we don't have acetylcholine, we're dead immediately because we can't move, nothing. Nothing happens. If we have no acetylcholine, but you inject somebody with epinephrine or adrenaline, they're still dead because you need acetylcholine to activate the presynaptic neuron that later releases the epinephrine. So acetylcholine is the stem of everything involved in the nervous system. Most important neurotransmitter. So we're going to learn all about that. And before that, the organization, general organization, and we're not going to cover in super detail about the different parts of the nervous system because we're just going to be talking about the main functions, the specific functions to sympathetic and parasympathetic, and then how they can be modulated by drugs. That's the ultimate goal of this class, is to learn about different drugs and the way they're modulating these pathways. So you have the central nervous system and the peripheral nervous system, right? Central nervous system is your brain and your spinal cord. Your peripheral nervous system is both your afferent nerves and your efferent nerves. So efferent nerves is everything stemming from your central nervous system out to your extremities. That includes your sympathetic and parasympathetic nerves, meaning do we want to increase activation of all of your limbs and extremities and organs, or do we want to decrease activity? Then your afferent neurons are the ones that are coming from your extremities, from all of your sensory abilities, and that information is getting fed through your spinal cord and goes to the brain for almost instantaneous reaction. So let's say if you put your hand over a hot flame, your afferent neurons are sending your nociceptors, those are pain receptors, are sending a signal saying, get your hand away. So it activates the skeletal muscle really quickly, which is a sympathetic nervous system response. Then blood is rushed to your muscles to move it away. That all happens in milliseconds. So it's pretty cool. Any questions on that so far? Can everybody online hear me pretty well? Or at least better than last week?
Yes.
Okay,
yes, we can hear you.
Good. Because I'm trying to hover over the thing. Okay. So we're going to be focusing on mainly the autonomic nervous system. So the efferent nerves, meaning everything that's going away from the spinal cord, away from the central nervous system, can be separated into two parts. Your motor neurons, which are different. They're a different type of receptor, the NM receptor, neuromuscular junction receptors. Those are different than your autonomic nervous system. What do you think autonomic means? What does it sound like? Automatic, right? That's exactly what it is. So your autonomic neurons, we don't control them. They are involuntary, which is why we need drugs to modulate them, because we can't do it ourselves. We don't need drugs to modulate our somatic motor neurons unless we have a disease in which we can't modulate them ourselves, like muscular dystrophy, for example. So our autonomic nervous system for everyone is something that we don't control. And there are two different parts of it: our sympathetic, which is our fight or flight, and our parasympathetic, which is our rest and digest. So parasympathetic, it says here, rest and digest, lacrimation, meaning crying, muscle growth, which means your protein synthesis. So a lot of the rest part of it, like let's say the reason why when you work out, rest and recovery is just as important as working out, is because your parasympathetic nervous system, when you're sleeping, you're doing your digesting, you're doing your protein synthesis of your muscles to all the broken down muscle to build new proteins on top of it. So that's important. And then for sympathetic nervous system, it's an immediate reaction. It has to do with an immediate danger or stress, right? So that includes sweating, increased heart rate, blood flow to your muscles, and decreased blood flow to your skin, which maybe makes you look flush. So that happens. And your sympathetic nervous system could activate when you're giving a presentation, and if you're not comfortable talking in front of people, right? Because you get all nervous and clammy maybe, and you start to feel a little bit weird, right? And that's your heart rate's increasing and all that stuff. So these are the two pathways we're going to modulate. Both parasympathetic and sympathetic nervous systems is you have two neurons for the most part. In the adrenal gland, you don't. You only have one neuron. But I'll show you that in the next slide. You have the central nervous system is the basis for all of your signaling, meaning that's where the ion gradients are created, and that's what propagates the cell body of the first, what we call the preganglionic neuron, or the presynaptic neuron, same thing. They are going to, that's where the signal originates from. That's where your action potential, you've heard of that term before, that's where your action potential is generated from. So that action potential will go along the axon, and by way of the ionic gradient, the electrical gradient, voltage gradient, generated by the myelin, in between the myelin sheath, the nodes of Ranvier. That's where the signal propagates along from. Which that part's not important for this class, but just to give you a little bit more. And basically, once that signal gets to the dendrites, the end of the synaptic cleft at the end, or the synaptic terminal. My bad. Dendrites are in the cell body. So we're right here. So once the signal gets there, that voltage gradient that has traveled along the entire length of the axon now activates what we call a synaptic vesicle. And I'll show you more detail later on, but this synaptic vesicle is what houses, it's a vesicle inside the cell that houses the neurotransmitters. Once that is activated, or the synaptic vesicle opens up, the neurotransmitters, acetylcholine, norepinephrine, dopamine, serotonin, those can be released into the synaptic cleft and can be taken up, and they can bind to receptors on the next cell, or the postganglionic neuron. That's what propagates the signal. Then on the postsynaptic neuron, we have another neurotransmitter that's released, and that one directly modulates your organs. Tells the organ, based on which receptor it binds to, it tells your organ to speed up or slow down. It's more complicated than that, but basically speed up or slow down. Parasympathetic being slow down, and sympathetic meaning speed up, for like muscles, right? So that's generally how it happens. So yeah, neurotransmitter. So what is a neurotransmitter, right? We didn't really talk about that. I just have mentioned a few. So it's a small molecule, meaning 300, or no, it's less than 300. It's like 200 or 200 to 300 Daltons. And they are released from either the presynaptic neuron or the postsynaptic neuron to activate the next part of the pathway, whether it be the presynaptic activating the postsynaptic, or the postsynaptic directly binding to the activator or the effector organ. So yeah, any questions on that concept? Same thing. Yeah, so that's the same thing. Preganglionic versus presynaptic neuron, same thing. Okay, good question. So something else. So the reason why I said acetylcholine is so important is because of our next slide, is that acetylcholine is involved in every type of nerve impulse, whether it be a parasympathetic response or a sympathetic response. So there's a lot going on here. And I'm going to break it down. And we have different types of receptors too, but we're gonna focus on this on a high level. So we're not going to go crazy deep into the different types of receptors. So we'll focus on the blue first, right? The blue, we have two pathways in blue. New laptop, you hear the fan? Here we go. Cool. All right, and the guys online, you can still see the screen, right? Oh, maybe not. Okay, now you should be able to. Okay, so in the blue, we have the parasympathetic nervous system types of neurotransmission. So what happens is you have your sensory output from the spinal cord goes along the preganglionic neuron or presynaptic neuron, and you have neurotransmitters are released. These neurotransmitters are acetylcholine. So all presynaptic neurons are acetylcholine. That's true. It's something you can write down. That's true for both the parasympathetic and sympathetic nervous system. It says it right down here. Acetylcholine released from all preganglionic fibers and stimulate NM or NN receptors on postganglionic cells. Now, that's true. So don't worry about the receptor type, the NN receptor. Just worry about that whether you have a sympathetic in red or a parasympathetic in blue, you have acetylcholine being released in the preganglionic neuron. Okay. Except for this middle case, doesn't have a preganglionic neuron, just has a post. But we'll talk about that in a second. So what happens on the parasympathetic nervous system signaling, these two are the same. Yeah, these two are the same. I don't know why they're both here. But anyway, so is you have these NN receptors. So N stands for nicotinic receptors. So they bind nicotine. That sounds familiar, right? So they bind nicotine. That's actually how these receptors were discovered. That they, when they characterizing them, they found out they bound nicotine, so they call them nicotinic. And muscarinic receptors are these, we call them M receptors. They bind muscarine, and they found that based on when they purified it, it bound muscarine. So these receptors are important. I mean, this is why nicotine is an addictive substance. It binds these nicotinic receptors all over the body, non-specifically, and directly modulates a euphoric feeling because you're getting that same as acetylcholine. You feel kind of like a depressing state, but it feels kind of good. So that's why it's such a nicotine is generally not a good thing because it can become addictive. So anyway, once these nicotinic receptors are activated, they then propagate the nerve in the action potential along the postganglionic neuron, which then bind or the synaptic cleft of that neuron is near the effector organ, which then it can release also acetylcholine, more acetylcholine in the parasympathetic case. So what this would do is we'll see a summary of the different types of physiological responses that acetylcholine generates. But again, they're all very depressive things. But it does stimulate digestion, right? Increased blood flow to the stomach, increased blood flow to, but increase their increases your increases dilation by decreasing constriction. So, but we'll see a whole list. But basically, it's a lot of functions that correlate to rest and digest. Just to keep it simple. So, any questions on that? That's parasympathetic. The way parasympathetic nerves work. Basically, acetylcholine throughout the whole way. Okay. Next is the red, sympathetic, right? So blue is cool, right? It's parasympathetic, rest and digest. Red is anger and blood and fire, right? So it's going to be stimulated when there's a really big stress. So this is, well, something I can mention. There's another, so over here, there's another type of nicotinic receptor. There's actually two. It's called NN and NM, right? M is for muscular. So the NM receptor is located on mostly smooth muscle. And this is how acetylcholine modulates increased contraction of the smooth muscle related to digestion. Because we have the postganglionic neuron is attached to the NM receptor on the smooth muscle, which then once the acetylcholine binds to NM receptors, it can stimulate the contraction of the smooth muscle. And these receptors, the NM receptor is a GPCR. So a lot of these are GPCR. Beta and alpha are GPCRs. The M1, M2, and M3, we'll talk about those later, but they are also GPCRs. So anyway, relating back to sympathetic. Sympathetic is fight or flight. So the first, like I said, the preganglionic neuron for all of them, parasympathetic and sympathetic, are modulated by acetylcholine. So acetylcholine is released, binds to the NN receptors, then the postganglionic neuron then releases your norepinephrine or epinephrine or adrenaline, whatever you want to call it. So norepinephrine and epinephrine are similar molecules. Norepinephrine is always around. So like the only difference between them, they're very simple and they do the same. They're very similar. They do the same thing. The only difference is that norepinephrine is always around at a basal level in the body, and it kind of creates the homeostasis between rest and digest, basically completely falling asleep versus at least a minimal amount of awakeness. Whereas epinephrine is only released from the adrenal gland in a moment of like, I need to act right now. But they both bind the same capacity to the same receptors. But epinephrine is just more of a quick release, and it's more of the that's why we would say epinephrine is the same. Epinephrine and adrenaline are the same thing. Norepinephrine is also called noradrenaline, depending on who you ask. Yes.
Is that because it's secreted more often, or is one cleared faster?
Good question. So I think epinephrine is cleared faster. It's very quick on, quick off.
Okay.
Well, yeah, good question. Okay, so norepinephrine and epinephrine are the main postganglionic neurotransmitters in the sympathetic nervous system that modulate all of your fight or flight, and they activate receptors called adrenergic receptors. So you have your alpha and beta adrenergic receptors, which we'll learn about. Okay, any questions on that? Okay. We have our two nervous systems. Now let's learn about the first one. Cholinergic. So the cholinergic system, no one needs to write all this down. We know what it is already. We kind of went over this. So cholinergic system composed of nerve cells that use neurotransmitter, acetylcholine, right? And they transduce action potentials, and they propagate nerve impulses, rest and digest. A cholinergic drug is a drug that binds to these cholinergic receptors. So basically, it's a drug that binds to an acetylcholine receptor. These NN receptors are NM receptors. So I'll put acetylcholine binds to NM, NN receptors. So it's any drug that can bind to those receptors also, and cause what also must be written. M1 through M3. It's any drug that can bind to those receptors to either inhibit it, enhance it, or mimic the action of the neurotransmitter acetylcholine. Anything that has a direct, and we'll learn a difference, but direct or indirect effect on the amount of acetylcholine, the reuptake of acetylcholine, or the modulation of the parasympathetic nervous system itself. That's what these cholinergic drugs do. And here's a couple of physiological conditions. So contract smooth muscle dilates blood vessels, meaning lower blood pressure, increased bodily secretions, so like urinating, defecating, and slows the heart rate. So we're talking about very depressive things. Physiologically depressive, not make you sad depressive. Okay. Any question on that? All right. Okay, so here's the different types of receptors. And I'm not going to, on an exam, I'm not going to ask you a crazy amount of detail on the receptors. Just know that there's, I would say, know that there's two different, you can even bunch these in two different types, your N receptors and your M receptors for acetylcholine. And then for, so it makes it easy. You have N and M for acetylcholine, for acetylcholine. Then for your norepinephrine, you have alpha and beta. So there's two types of receptors for each one. So yeah, so your N receptors, they're on their postganglionic neurons, and they're activated by acetylcholine, like we saw, and that's what activates the postganglionic neuron. And your M receptors, remember the M stands for neuromuscular, muscular. Think of it as muscular. So they're on the neuromuscular junctions, and also they control smooth muscle and also skeletal muscle. And again, don't go crazy on the detail of this. Just know that there's two types. And then muscarinic receptors, there's three types: M1, M2, M3, and they are in the postganglionic neuron, and they are the one that secretes or releases your acetylcholine onto whatever effector organ there are. So whether it be the heart to slow down the heart rate, the smooth muscle to increase contraction, or some of the lungs or the blood vessels to dilate them, your eyes to constrict them. So that's another example of something that is constricted or contracted. Your ocular muscles are contracted, so your eyes are constricted when your parasympathetic nervous system takes into or is in action. Because when your sympathetic nervous system's in action, your eyes dilate. So you can take in more light, see more, and react better. Yes. No. So just know that there's two. For the parasympathetic nervous system, there's your N receptors. But just don't need to know this. You just need to know there's the N receptors, then there's the M receptors. And then for the sympathetic nervous system, we'll talk about in detail in the second half. You have your alpha and your beta, right? And there's two types. There's actually two. There's alpha 1 and alpha 2. They're very similar. There's beta 1 and beta 2. They're not as similar. They have specific functions, but that'll be easy to learn. So I'll tell you right now. Beta 1, what do you have one of? Heart, exactly. So beta 1 is your heart. So if it's activated, increasing heart rate. Beta 2, what do you have two of? Lungs, right? So beta 2, when they're activated, increases respiratory rate. That's basically it. That's a quick little lesson. Okay. So here's a big summary. We kind of went over this already in little snippets. If you want everything in kind of one place, you have your cholinergic responses, meaning your parasympathetic, your NM and NN receptors, you have acetylcholine, and then you have more acetylcholine in the postganglionic neuron, unless you have your adrenergic system, which in case you have norepinephrine. That's basically how it works. This is from a YouTube video, as you can tell. I snipped out. I think it's in the, it's plagiarism. It's not in the notes. Should be. I always do that, but okay, I guess I forgot. Anyway, any questions? Okay. So let's take a look at some comparisons. That's blurry. Let's take a look at some comparisons between the different nervous systems real quick. So sympathetic nervous system. So this is a good question to ask. The sympathetic nervous system, its origin is the central nervous system. They're both central nervous, but the sympathetic nervous system originates from the spine. The parasympathetic nervous system originates from the brain. Why is that? Why would we need the sympathetic nervous system to be in the spinal cord? Or why is it better than it is to be in the brain? quicker, right? Exactly. So evolution, as crazy as it works, over time, let's say there were some organisms that had their sympathetic nervous system closer to their brain, their response times to danger were split seconds less, which caused them over millions of years to die off. And the species that had a sympathetic nervous system in their spine, which is closer to our limbs physically, and we can propagate impulses faster, they survived more because they can react quicker to danger. That's pretty cool. So yeah. Okay, also length of fibers. So same idea with the length of the fibers. The preganglionic neuron is really short because we want to use that. We want to propagate the impulse by acetylcholine quick. But then when it comes to your postganglionic neuron, that signal propagates, and then you have norepinephrine. And that norepinephrine, it has to reach your limbs. So the length is going to be a lot longer. And it's going to be really, you're releasing norepinephrine all over your different extremities. So there's a varying length of the neuron. Location of ganglia, talked about that already. Preganglionic fiber branching, extensive. So the branching, again, we have to reach receptors instantaneously or really, really fast of all over because we want to move really quick. Distribution, type of response, diffusive, discrete. Okay, so yeah, any questions on that? All right, so more importantly, why do we care, right? So we care about this because knowing the different receptors that bind acetylcholine and norepinephrine or epinephrine, we can make drugs to modulate their signal. So that's basically it. We can either directly or indirect. And the things we can modulate are extensive. So here's, I'm not going to go over every single thing, but we went over most of this already, verbally. The idea of sympathetic versus parasympathetic, I've said it 100 times already, so I'm not going to really go crazy, but you can see how, and you can take a look at this if you want later, but you have the different nervous systems involved, right? Blue, you have parasympathetic, red, you have sympathetic, the receptor type, and then what it does, right? So you can theoretically, with a drug that activates this pathway or activates this receptor or inhibits this receptor, you can inhibit these different physiological conditions, which is extremely important. We have blood pressure is a huge part, bronchoconstriction, bronchodilation, intestinal motion, retention in terms of like waste retention, ocular pressure, and stuff like that. So these are really important physiological modalities that we can change with manipulation or small molecule manipulation of these different receptors. Okay, so next question is, Professor, how do I do that? Well, you first need to know about molecularly what's going on. Now, we talked about in big detail, and this is where you don't need to memorize this, but I may intend to give you on an exam a diagram, and you might have to answer, what are 3 ways we can prevent this from happening? Or you have a condition that prevents this from happening. What are 3 ways we can start this happening again? Or what are 3 ways we can propagate the neurotransmitters? Sorry, it's Friday night. I'm just like, but we're all together. We're all doing it. All right. So this process is similar for both acetylcholine and norepinephrine, where you have the synaptic cleft, which is over here, the synaptic vesicle, which is here, and these blue things are the neurotransmitter. Okay, so I'll walk you through it. We'll start at number 1. Number 1, synthesis of acetylcholine. You have the choline acetyltransferase. Don't worry about the enzymes too much. We're not going to focus on that. But you have choline acetyltransferase, which is an enzyme that takes acetylcholine or makes acetylcholine from choline and acetyl coenzyme A. And you get acetylcholine. So right off the bat, even from step one, you can figure out a way that if you had too much acetylcholine, how do we stop it? You can answer this question. So how would we, based on the first step alone, what can we do? What can we make? Which? Well, that's too far. So just looking at number one. So in that one sentence, it says choline acetyltransferase catalyzes the synthesis of acetylcholine from choline and acetyl-CoA. But what can we do to stop that? Exactly. Yes. We can have a choline acetyltransferase inhibitor, and that will block the process from happening. Nothing else would happen. You'd also die because it would be non-specific and it would happen all throughout the body. So another concept, in general drug discovery concept, the further you modulate down in the pathway, the more specific it is. Because a lot of these pathways, they branch off into different things. So if you can take one of the if you can modulate and inhibit one of the enzymes that's like down here rather than up there, it'll be more specific. But it's also more difficult to do that. So it's what's easier, to drug a receptor or to drug one of the proteins down below that one of the transcription factors. The receptor is easier to drug, but the receptor also controls all that stuff, which can lead to a lot of side effects. That's one of the biggest troubles of drug discovery, is how do we get down there, right? We need a drug that is lipophilic enough to diffuse through the membrane, which is tough alone. And you need one that won't be metabolized and will have function once it gets down there. That's a lot of obstacles already. But anyway, let's go back to this. All right, so number two, uptake into the storage vesicle. So acetylcholine gets stored in this synaptic vesicle. Technically, you can inhibit that. You can find a drug that breaks down the vesicle membrane. Sure, but I mean, I don't think that's really a widely used approach, but that would be correct on an exam if you could explain it a little bit more. If you had an idea, right? So then once you have stimulation by the action potential, right? Action potential comes through, calcium is influxed through the calcium ion channel, which then leads to depolarization. Depolarization allows the synaptic vesicle to open at a certain voltage, and then neurotransmitters are released into the synaptic cleft. Now, in the synaptic cleft, they bind to their receptor, their postganglionic receptor, or their receptor on whatever organ they're on to cause that response. But that's not the end of the story. We also have acetylcholine just chilling, just sitting in the synaptic cleft. What happens to it? Well, there's a degradation process for acetylcholine. Same thing for every other neurotransmitter too. So for acetylcholine, it gets broken down by acetylcholine esterase. It cleaves the ester bond of acetylcholine. And a drug class, they have acetylcholine esterase inhibitors. One of those is called cocaine. So, well, it's not specific. It's a dopamine reuptake inhibitor, serotonin reuptake inhibitor, and acetylcholine reuptake inhibitor. So to different extents. Mainly, I kind of misspoke. Mostly dopamine and serotonin, but also acetylcholine. Because the enzymes are similar. So that is a way to increase acetylcholine, is by destroying or inhibiting the enzyme that breaks it down. What that does is it prolongs the maximum concentration, or prolongs the time that the maximum concentration is present in the synaptic cleft, allowing more binding, a longer propagation of this signaling, whereas maybe it would happen for 10 minutes, it would happen for 10 hours, right? So destroy or inhibiting the enzyme that breaks down the neurotransmitter is a great way to activate the neurotransmitter or prolong its activity. This is called indirect stimulation. You can directly stimulate acetylcholine by increasing by binding an activator to the receptor, for example, right? Something that will change the shape of the receptor ever so slightly, that it has a higher affinity for acetylcholine, right? or by increasing the activation of acetylcholine transfer or acetyltransferase, meaning you make more acetylcholine. That's direct. Indirect is by decreasing its uptake. So that's something to think about when we're talking about taking a diagram, finding different ways to modulate it. So just to think about which enzymes can I attack. You notice all my targets that I've said are all enzymes, because those are the things that are druggable. You can't really drug a membrane, right? That doesn't really make any sense. Unless you're talking about antibiotics, in case like penicillin breaks down the cell wall, kind of drugging the membrane, but you're still targeting peptides that are on the membrane. So you always have to target a protein, because you can drug it a lot better, because you can figure out a structure. Okay. Also, you have receptors that are located on the presynaptic neuron that bind acetylcholine. So after acetylcholine is released, you have receptors that also bind it on the neuron that already released them. Why would we have that? What's those receptors' purpose? The opposite of that. Stop it. Yeah, right. So we can have a signal that starts from acetylcholine binding to those presynaptic receptors that causes a negative feedback. So that's telling the neuron, all right, don't release any more acetylcholine, because we already have enough. We have so much that some of it actually rebounds to the previous neuron. So another way to activate the amount of acetylcholine, or increase the amount of acetylcholine, is to make an inhibitor for this receptor. So there's a lot of ways. Cool. Good question. They don't. So this receptor has a higher affinity than this one. This one might be, and since we're this is a natural ligand, we're talking on the scale of probably high picomolar, and this one's probably high nanomolar. So yeah, or mid nanomolar. I'm not exactly sure. It varies, but in that range. So yeah, cool. Any questions? Okay. All right, so let's go through an example. So we have pilocarpine is a muscarinic. Don't worry about the type of cholinergic drug it is, but it's a muscarinic agonist. So it binds to one of the acetylcholine receptors, and it's an agonist, meaning it activates it. Okay. So what is it used for? It's a non-selective agonist that binds to, it's used to treat glaucoma and xeristomia, which is salivary glands don't produce enough saliva. Basically means your mouth is dry all the time. And it's non-selective. What that means is there's 3 types of muscarinic receptors, M1 through M3. It binds all of them, which can lead to a lot of side effects, right? Off-target effects. So they've been developing muscarinic or specific muscarinic agonists. That's difficult because all 3 muscarinic receptors, even though they're located in different places of the body, they're both pretty homogeneous. They're both complementary to some extent. Yeah, so that's one example. So it also is used to treat glaucoma. So which is you have an increased eye pressure. So what the drug does is it stimulates the ciliary and iris muscles contraction, which then decreases the optical pressure. So basically, it works as a agonist, which means it's a acetylcholine mimetic. It activates that pathway where acetylcholine is not there. This drug, pilocarpine, will bind there instead and activate the same pathway. So yeah, that's one example. Another example is you have the other type of receptors, nicotinic. You have a nicotinic agonist. So next slide. I put a drug here. But anyway, so it's not important. It's too much. It's more specifics on the nicotinic receptor. But basically, nicotinic receptors are ligand-gated ion channels. So they're not GPCRs. The muscarinics are GPCRs. The nicotinics are mostly ligand-gated ion channels, which they bind acetylcholine, and then they allow sodium to enter the cell, which then can propagate the action potential. So they bind nicotine, but they can also bind other drugs that we make to agonize them. And there will be some examples. Here we go. So anyway, so here are some drugs that are direct acting. So we talk about indirect acting, which means they bind to, let's say they inhibit the degradation, right? That's indirect acting. But direct acting is they bind, like pilocarpine is direct acting because it binds to the receptor and activates it. So whereas, even though it's nonspecific and direct. So here's some other direct ones. You have carbachol and bethanachol. So they are cholines, and they're similar to acetylcholine in structure. And what they do is they bind to the nicotinic receptor, and they act as an acetylcholine molecule, right? They're mimetics. And then we have the nicotine and nicotine binds to nicotinic receptors, obviously, but nicotine can also bind to muscarinic receptors. So, but not as much of a affinity for them as they do for nicotinic receptors, obviously. But pilocarpine and bethanachol, they bind to muscarinic receptors, and they can be called muscarinic agents. So basically, we call it two types of drugs, but literally they're all very similar in structure, having the same basic structure or same core as acetylcholine, but they are more selective to either nicotinic or muscarinic, depending on the type, depending on the drug. So here's a good question. So muscarinic receptors are GPCRs, right? They look completely different than ligand-gated ion channels. Nicotinic receptors are ligand-gated ion channels, right? So you have GPCR, which looks like that in the membrane. And then you have ion channel, which may look like that with like a channel going through it. And then you have binding sites over here, right? So they both bind acetylcholine. How's that possible? They look completely different. How can they both bind acetylcholine? It's not as complicated of an answer as you think. What do you mean? Close. So yeah. So the entire protein is not responsible for binding, right? Just the binding site. The binding sites are pretty small. So the complementarity between this binding site where acetylcholine goes and the one on the GPCR where acetylcholine goes, they are extremely similar in sequence. The rest of the protein is completely different, but the binding sites are conserved. This is true for, you know how many ATPase proteins there are? Proteins that use ATP for energy, right? A ton. And they're all completely different. There's so many ATP binding proteins. And the rest of the protein, they might have different domains, or different cell types, different tissues, different species of animal, right? But they all have the same or very similar ATP binding site. So that's true for a lot of drugs. They have similar receptor sites, but the entire protein is different. So it's good to know. Okay, so we talked about this already, but let's just review it again. If we're talking about indirect acting cholinomimetics, I know it's a mouthful. Cholinomimetics meaning cholinergic mimetic, something that acts like acetylcholine, but they're indirectly acting. So they do the same thing, even though they're called, I don't really agree with the term cholinomimetics, because that means it's a molecule that's like acetylcholine. But even though these molecules may not be like acetylcholine, they still produce the same physiological response as acetylcholine, which is why they're called mimetics, and they can get away with that. So what we can do to indirectly act on cholinomimetics, or indirectly act on the cholinergic nervous system, is we can inhibit the destruction of acetylcholine. We talked about that either by the acetylcholine esterase enzyme inhibiting it, or by competitively inhibiting the receptor on the presynaptic neuron, or by the inhibition of the enzyme, what enzyme? Choline transfer. What was it? What enzyme was it? The acetyltransferase. Yes, the acetyltransferase enzyme. Right. We can inhibit that, or sorry, we can activate that to make more acetylcholine. So yeah, those are ways. They can be reversible, or they can be irreversible. So we didn't really talk about reversible versus irreversible drugs. So for most cases, irreversible is bad. Why is irreversible bad? Right. So yeah, basically, yeah. So you can't stop the response that it causes. And we mean by irreversible, we mean covalent. So a lot of times covalent inhibitors, they make, so really the way drugs, we don't really talk about drug binding. We should, we will. You know what? Remember I showed you that list of like what we're gonna do in week 12 was like a question mark. We'll do that. We'll talk about like more specific, like the molecules of like drug binding, like intermolecular forces, right? So all these drugs that we learn about that are FDA approved they use intermolecular forces, Van der Waals equations, Van der Waals equation. Wow, I'm losing my mind. Van der Waals interactions, some ion-ion interactions. Hydrogen bonds are a big part of it, right? But they're not covalent. They're covalent. It takes a ton of energy for it to come off, and in the body, it's not going to come off. So we don't want that. We don't want these signals to be propagating for that long. So in order to, well, I'll ask you guys a question. So if we have an irreversible drug, how do we survive? How do we stop the signaling of that drug? Potential, no, because it's bound. It's covalently bound, so it's not gonna. But if we had a reversible drug that, even if it has like a picomolar affinity, if you dump in enough of nanomolar affinity drug that competes with it, it will compete it out. Combining that with metabolism and everything, it will compete out. But if you have drugs that, even if it's not that potent and is irreversible, that's bad. What can you do to what can happen? Or what do you need to happen? That's actually a closer answer than to the real answer. Yeah. So basically, you have to wait until. So okay, think of the cell as a human body, right? We are constantly going through mitosis in all of our cells. Our skin regenerates after like 4 weeks completely. All the skin cells are different. So in that process, going down to the cellular level, you have proteins that are being broken down by the proteasome, by the lysosome, and then receptors or proteins being resynthesized by protein synthesis and by gene expression. So what you have to do if you have nerve gas inhalation and you are in the hospital and you can't, and you're like, they have paralysis, what they have to do is they have to give you enough nutrients and just keep you on a ventilator until your cells go through mitosis, recycle those receptors, and replace them with new receptors, which can take up to a week, depending on which receptor. So that's really the only treatment for irreversible inhibitors. So just to keep you alive enough until your body fixes it. Yeah, and in some severe cases, yes. But in these kind of processes, you know that you know what's going to happen. So like, I mean, usually you see this happen with people that are involved with like pesticides or different types of agricultural plants where they're constantly exposed to this stuff, but it's somewhat controlled. It's not gonna be chemical warfare, because if that was the case, you don't have access to life support most of the time. You die on the battlefield because you can't breathe after a while, so you can't breathe and that's it. But yeah, so that's basically, it's scary, irreversible inhibitors. So yeah. Okay, cool. Any questions? So there are some reversible anti-cholinesterase inhibitors. That should say acetyl. Yeah. Did I write? I don't think I wrote that. I got that from somewhere. I wouldn't write that. That doesn't make good sense. It should be acetylcholinesterase. Okay. So anyway, you have endorphin, which isn't really that really use it that much. But these other ones, we have a bunch of these drugs that what they do is they do exactly that. They are indirectly increasing the amount of acetylcholine by inhibiting acetylcholinesterase. That's the anti-cholinesterase is bothered. So it's acetylcholinesterase. And yeah, they can be used for different types of diseases, like myasthenia gravis. So basically, what that is it's an autoimmune disease. So your body destroys or recycles your NM receptors. So on the neuromuscular junctions of your smooth muscle and your skeletal muscle, you have no receptors to bind acetylcholine, or very small. The receptor amount is limited. So if that's the case, then you need to prolong the amount of acetylcholine release and response to get to those few receptors in order to be able to contract your muscles. And it's not a great treatment, but it's something that improves the condition. And it gets worse over time, that kind of disease. So, a couple of things that are important. Alzheimer's disease, one of the biggest targets right now in drug discovery. So something that we notice with Alzheimer's disease is that it's correlated to a very low acetylcholine level. Why do you think that is? The idea. Simple answer. So in the brain, what's acetylcholine doing? Sending signals. If you have not much of it, it's not sending many signals. And then memory, right? You forget things and you end up living a bit differently, right? A lot slower. So that's one of the conditions of having Alzheimer's is low acetylcholine, among other things, beta amyloid plaques and other things like that. But you have low acetylcholine. Now the question is for someone. So drug discovery, we want to think about it really simple. All right, if you have low acetylcholine in somebody with Alzheimer's, why can't you treat them with acetylcholine? What do you guys think? Why can't you treat them with acetylcholine? That's one answer. It's not specific. What? Oh, so why can't you said what was the question? Oh, so why can't you treat, since Alzheimer's is correlated to having low acetylcholine? What if you increase those neural connections by increasing the amount of acetylcholine and just giving it to them? Say it again? Exactly. So that's the better answer, but that's also right. But so like let's say it could pass the blood-brain barrier, it would work, but you'd also have a lot of non-specific effects, right? But the main reason why, no matter how much you give, it wouldn't work is because acetylcholine is pretty polar compared to something that's non-polar enough to cross the blood-brain barrier. So it can't cross at all. That's why you can't use it for a psychoactive drug or anything in the brain. Good. So then when we have another disease, Parkinson's is another disease that's of major interest in drug discovery world. And it's represented by high acetylcholine, right? You have a lot of neural connections. They're kind of dysfunctional neural connections, and you end up with someone that has tremors and different kinds of involuntary movements created by the increased amount of acetylcholine. So different drugs you can treat Parkinson's with is, yes. So a dopamine reuptake inhibitor, that would work. because it also corresponds to low dopamine, right? Good. Also, you can activate your acetylcholinesterase inhibitor. But no, your acetylcholinesterase. Yes. Because then it would break down more acetylcholine. So there's different ways to attack it. And all these different things that we're saying are being tried by a lot of pharmas around the world. So, yeah. Right. So that's a common thing. That's a good question. We see that a lot in like where marijuana as a treatment for Parkinson's. It's still under debate. Nobody really knows the true mechanisms. That the cannabinoid receptors, CB1 and CB2 receptors, are still highly studied. We don't know too much yet. People know more than me, definitely, people who are on the cutting edge of it, but I don't know too much, and the general science community doesn't know that much, but some experts in it obviously do. So, I mean, feel free to look up papers on that, but there is some, I believe there is some beneficial properties to it. So, definitely. Of course. Right. Also depends on the different molecules, the different THC derivatives out there. A lot of them. So, and they all act a little bit differently. So it's basically you're drugging the cannabinoid receptor in such a way where it's specific enough and it creates what the desired response. And that all relates to the pharmacokinetics and then the responder, the drug concentration versus response graphs, right? So it all relates to that and how much of a physiological outcome is there, how much of a response. So all good stuff. All right, so we have irreversible inhibitors. We talked about this with like organophosphates. So that's like nerve gas. And basically you have to wait until the receptors regenerate themselves. Yeah, so restoration of the acetylcholine esterase activity requires synthesis of new enzyme molecules. So you need to just wait for that synthesis. Okay, so next thing is cholinergic agonists. We're about halfway through, so we'll be fine. And then, yeah, so the adrenergic system is going to be quicker. So cholinergic antagonists. So in this case, we have a condition where we have too much acetylcholine and we want to stop it. So let's say we have Parkinson's, right? There are different ways to treat Parkinson's with modulating acetylcholine, but dopamine is one way to modulate it. But you can have different antagonists. So these can be competitive inhibitors, meaning they bind where acetylcholine would bind to the muscarinic or the nicotinic receptor, and they don't allow acetylcholine to bind. And their structure is not really too much like acetylcholine to where they will propagate that signal and cause an activation of that receptor. They will actually inhibit the signal. That's possible too. So drugs like this, you have atropine. Atropine is the main one, so we can talk about that one. So here is also scopolamine, but atropine is like the main one. So it's a competitive blocker, competitive inhibitor of the muscarinic receptor. And here's basically how it works. So you have acetylcholine would normally bind to either one of these drugs will bind to the muscarinic receptor with, in terms of affinities, it's less most of the time. Acetylcholine is more has a higher affinity, but treating with enough of atropine would definitely compete out some of the acetylcholine. But it has to be a high enough dosage. So, right, so they all fit into the receptor. So it's a part of it, what we call the I guess we could use the term warhead. I want to use the term. I've heard it like so many times today. I just want to use it. So the warhead, meaning the part of the molecule that coordinates the binding into the binding pocket, we call it the warhead sometimes. So that part rest of the molecule is what's blocking or making additional interactions to not cause the muscarinic receptor to be activated. It just leaves it in just a blocked state, right? It's like putting, you ever seen the basketball hoops at like sports, well, sports authority, but like Models where they have the glass thing on top? When I was little, I used to try to shoot it in. It's like that. That's what a competitive inhibitor does. So it's the same shape, right? But it's not going to allow the natural acetylcholine to get in there, you know, same thing. Cool. Yeah, so when so atropine, what it's used for is when you have low heart rate. So bradycardia, meaning low heart rate. Tachycardia means high heart rate. Now, low heart rate, how that makes sense because you have atropine. To lower the heart rate, you need acetylcholine lowers the heart rate. So if you competitively kick out acetylcholine, you're going to have an increased heart rate because you're going to have a less parasympathetic activity. So that's a direct inhibition. There's also other things that can happen. You could use it to reduce salivation, and other things. Mushroom poisoning, can't go for the overdose. So here's a question down here. So I don't know if I want this. So pyridostigmine, it's an acetylcholine esterase inhibitor, right? Why can it be used in the overdose of atropine? What does it do? It binds, it's a competitive inhibitor with acetylcholine, with the muscarinic receptor, decreasing the amount of acetylcholine. Why can an acetylcholine esterase inhibitor be used to treat an overdose? Right. Yes, yes. So it's actually a dual mechanism. So that's one of the answers. So what will happen is the acetylcholine that's in the synaptic cleft will not get will not get broken down, right? Therefore, some of it it would increase the effective concentration in order to one bind back, like you said, bind to the muscarinic receptor, restoring activity. Also, it increases the effective concentration of acetylcholine for downstream effects. So it will preserve the downstream signaling of acetylcholine, even though maybe the muscarinic receptor on the same neuron is inhibited. So it can work in both ways. And then until we get all that drug out, that can be used to decrease the side effects of that, or decrease that toxicity. Okay. Any questions on cholinergic? Okay. Now it's getting late. All right. Adrenergic system. So fight or flight. Now, the first half of the class we talked about this. Acetylcholine modulated, right? You have two types of receptors, nicotinic and muscarinic. And we talked about the different types. Briefly, we talked about different types of competitive. Competitive meaning direct. We have indirect. We have antagonists, and we have agonists. Now, the adrenergic system, there's basically also two types of receptors. There's alpha receptors and beta receptors. There's alpha 1, alpha 2, beta 1, beta 2. Now, let's go through some key terms before we start. So epinephrine, norepinephrine. So they're, like we said, they're similar. Epinephrine is actually synthesized from norepinephrine, and it's released from the adrenal gland. Norepinephrine binds, and they both bind to alpha and beta adrenergic receptors, but norepinephrine binds to alpha better than it binds to beta. Not really too important for the exams, just to know. And we have both of these. We can consider them to be similar. Just the epinephrine gets released from the adrenal gland in a quick response to stress, and is released and is degraded quicker. Then we have the alpha-1 receptor. So the alpha-1 receptor is a G-alpha-Q GPCR. We talked about G-alpha-S and G-alpha-I. There's another type called G-alpha-Q, which it follows a similar pathway. It's an activation pathway, obviously, because we're talking about adrenergic. But it's a little bit different than the adenylyl cyclase kind of pathway. It follows, we didn't really go over it, but there's molecules called PIP2, IP3, and DAG, which basically over time it increases the amount of calcium in the cell. We need calcium for muscle constriction. So that's what it does. So that's the alpha-1. Alpha-2 is a G-alpha-I, right? We know about G-alpha-I. What does G-alpha-I do? Inhibits. Good. G-alpha-I for inhibition, right? So it inhibits the release of norepinephrine. So it does the opposite of what the adrenergic system should do. It decreases the amount of fight or flight response. Beta receptors, you have their G-alpha-S receptors. They're a canonical activation type of receptor. They increase your heart rate, they increase hormone release, bronchodilation, vasodilation, smooth muscle relaxation, meaning the stomach, you're going to have less digestion. So something when I did track in high school, you would have the kids that would eat at, like, let's say track practice was at 2:30. The kids that would eat lunch at 2 o'clock, they're most likely to throw up. Because when you're sprinting in the freezing cold, your adrenergic system is on fire, right? So all of your blood is going to be diverted from your smooth muscle, from your digestive system. Your smooth muscles are going to relax. The blood's going to be away from it. So then you have all this food jumping around, and there's not really many enzymes or blood to break it down or to control those processes to break it down. And then it's more likely to come up. And you just feel, your stomach doesn't feel good. So definitely eat way before you exercise because of this reason. So yeah, any questions on the two types of receptors? Yeah, so for the exam, for alpha, it would be good if you knew alpha-1, alpha-2. Alpha-1 was our general activates like all the sympathetic nervous system. Alpha-2 is an inhibitor one, so it's a G-alpha-I. That's an important difference. Beta receptors, if you can remember beta-1 and beta-2, beta-1 mainly for heart, beta-2 for lungs, that would be good also. So I got an open-ended question. If you added that, the points would be in your favor more. So like I wouldn't take off points for not knowing that, but I may take off points. I may add points for knowing. So it's good to know. Okay, so we kind of talked about all this already. In terms of drugs, it's the same type of drugs. You have, instead of parasympathetic mimetics, you have sympathomimetics and what we call sympatholytics. Sympathomimetics, what do you think? Something that looks like norepinephrine or looks like epinephrine, and it activates the same pathway. Sympatholytics, lysis, lytic, breaking down, right? Bad. No, right? So it's something that decreases the activation of the adrenergic system. These sympathomimetics or sympatholytics could be indirect or direct. Same kind of concept. I'm going to write this down. It's important. Okay. So this kind of breaks down, hopefully you guys online can see. So this kind of breaks down everything we're talking about. Different classifications of nervous system. They can either be indirect or direct or indirect. They could be sympathomimetics, meaning something that's like norepinephrine or epinephrine, stimulates. Sympatholytics, something that does not stimulate the sympathetic nervous system. Cholinomimetics, these are similar. Sympatholytics and cholinomimetics are similar physiological outcomes because sympatholytics, meaning you're not activating the adrenergic system, which is you're not activating fight or flight. Cholinomimetics, you are activating rest digest. They're kind of the same thing in terms of the outcome, but they're completely different drugs that work on completely different receptor systems. So important. Cholinergics are not going to be the same as sympathomimetics. They're not going to have the same outcome. Sympathomimetics, all your pressures are rising and you're ready to run or fight. Cholinergics, you are just decreasing the effect of the acetylcholine, not necessarily increasing the effect of your adrenergic receptor. So I'm going to put a little bracket around these. And either way, these things can happen indirectly, meaning they are binding to receptors that are associated with the reuptake or the synthesis of acetylcholine or norepinephrine, or they could be direct, where they bind to the exact receptors competitively as acetylcholine or norepinephrine. Does that make a little more sense? Because I feel like I'm just rattling off different concepts, but we want to organize it a little bit better. This is how we organize it of different types of drugs. So if I'm saying in text, and I've seen this before, we can kind of like think about the words. If I say we have a indirect sympathomimetic, and you're like, whoa, what do those words mean, right? So you break it down. Indirect, right? That means okay. It's not binding to the sympathetic nervous system, meaning alpha or beta, but it's mimicking it, meaning it's causing it to increase, not by binding to those receptors. Oh, how else can it increase? Oh, you know, it'd be like A, B, C, and D. It can increase by binding to the reuptake inhibitor. Yep, that's the answer, because that's indirect. So questions like that would be fair game. And then, so you have to kind of like walk through the words and see what it means, because you know what those words mean individually. Once I put them together, it gets a little bit more complicated, but you can also break it down and figure it out. Okay. So does that make sense? Okay, cool. Yeah, so norepinephrine, we talked about it already, constantly released. Epinephrine only released in times of stress from the adrenal medulla. Yeah. Okay, so look at this. This looks familiar, right? Very similar to acetylcholine, but it's norepinephrine. The synthesis and the enzymes are a little bit different, but the process is the same. So synthesis of norepinephrine, you have what I'm going to cover. We have dopamine and dopamine. So dopamine is actually the precursor for norepinephrine. So dopamine is covered in the storage process. Dopamine converts into and then after it's converted, we have the same thing, the release, the binding to the postsynaptic neuron, or in this case, the whatever organ it is, right? Because we're not dealing with preganglionic neurons anymore. We're dealing with strictly the organ or the place of action, right? So you're going to have binding to that receptor of that place of action, meaning alpha or beta. And you're going to have maybe some binding to the presynaptic receptor to cause the negative feedback loop. And you're also going to have degradation and metabolism. So the degradation, you have a different type of enzyme called catechol methyltransferase. CHEM-777, it's called. So right here, catechol methyltransferase. What does it do? You just read the name of it, methyltransferase. What is it? O-methyltransferase. What does it do? Yep, exactly. So it transfers a methyl group to the catechol, right? A catecholamine is what norepinephrine is, and epinephrine, catecholamine, right? So it transfers a methyl to the catecholamine, making it an inactive metabolite. Then that can be metabolized and put into the urine and excreted. So that's one way to indirectly modulate norepinephrine. You can activate norepinephrine by inhibiting catechol O-methyltransferase. Yeah. So you have another enzyme called MAO, or monoamine oxidase. That's another enzyme that does the same thing. It reduces norepinephrine and it makes it a metabolite and breaks it down, or conjugates by what oxidizes it. So that's another enzyme that could be targeted as an indirect adrenergic agonist or antagonist, but indirect. If we want it to be direct, we can target this receptor or this receptor, or dopamine. We can have a dopamine blocker, which would block the synthesis of norepinephrine. So yeah, but in general, the pathway looks similar, and we just have to think of what are ways we can stop it, what are ways we can start it. Just based on that, if you were given a diagram without the words on it. Or I'll include these. Yeah. Also, you have serotonin norepinephrine reuptake inhibitor. So those are other drugs. That's like cocaine does that. Okay. Any questions on that? Yeah. So cocaine, it is a serotonin norepinephrine reuptake inhibitor. So what it does, it is, so I'll use this word, I'll use one of the terms. It is an indirect adrenergic agonist. So indirect, meaning it doesn't directly bind. Binds to. Adrenergic means it's responsible in the adrenergic system, fight or flight. What did I say? Inhibitor? No, what I said? No, active agonist. I forgot what I said. Agonist, meaning it activates your sympathetic nervous system, right? People who you're on coke, right? That means you're just energied, you're all over the place, all this stuff, right? You're hyperstimulated. That is from the norepinephrine decreased reuptake, meaning there's more epinephrine in the synaptic cleft than there normally would be, and it's there for a prolonged period of time. People who do coke, you could be up all night, right, from one thing of coke, one line or whatever, right? And it really ruins your life, and also compounded with the addiction, and then you can die. So it's really not good. So but yeah, that's how it works. To ADHD, so yes, in essence, we're actually gonna. It's interesting you mentioned that. So we're gonna talk about Ritalin. Where's Ritalin? Are I talking about Ritalin? Yes. So Ritalin is a drug used for people with ADHD, and what it does is it's called methylphenidate. It inhibits the dopamine transport. So it works in a different way. What we can do, so dopamine increases your focus. It also makes you happier. Dopamine's good generally, right? But dopamine can also cause addiction. So if you have like the opiates, they induce an increased amount of dopamine, which leads to that physiological or that psychological addiction. So it makes you feel good, right? But dopamine is also responsible for attentiveness and focus, right? Cocaine is the opposite. You're kind of focused on everything at the same time. It's not really focusedness. It's just you're just hyperstimulated. So for a drug to treat ADHD, we use methylphenidate to inhibit the transport or the reuptake of dopamine, meaning you're more attentive and you can focus better. So yeah, it's good that you mentioned that. Okay, cool. So let's go here. So we're not going to focus on this really, but if you want to know more about which specific receptors for the adrenergic system that the catecholamines act on, they all have some affinity to all of them except for isoproterenol. That's a very uncommon one to talk about, an uncommon adrenergic catecholamine. But so anyway, the catecholamine, what it is, it's going to break down the word catechol, which is a catechol is a benzene with 2 hydroxyls on it, cholamine. So that's what a catecholamine is. And derivatives of catecholamine, we have epinephrine, norepinephrine, isoproterenol, dopamine, and dobutamine. And they all act on all or a combination of the receptors, the adrenergic receptors. So we're not going to go crazy on them. Epinephrine acts on all of them. It's non-specific. We need everything to fire at the same time. Norepinephrine, mostly alpha, more than beta. Because if it acted on beta, right? If it was a high affinity towards beta, what does beta control? Beta 1 controls your heart. Beta 2 controls your lungs. It's better for us physiologically to not have a constantly available neurotransmitter that's increasing our heart rate and our respiratory rate all the time. So the fact that it has a lower affinity for the beta 2 and beta 1 is good compared to alpha 1. Yeah, dopamine is mainly beta 1, and dobutamine is mainly beta 1 also. Yeah, those are lower affinities too. So if you wanted just more information on the different types of receptors, alpha 1, alpha 2, beta 1, beta 2, here's more information. So the alpha 2, remember we talked about alpha 1 versus alpha 2. Alpha 1 is the G alpha Q, meaning it's a different pathway than G alpha S, but it's still an I'm about to say activatory, that's not a word, an activation mechanism. But alpha 2 is G alpha I, which is inhibition. So a good question, which we know the answer to, is why do we need receptors that bind the same ligand that produce the opposite response? So who wants to answer that? Oh yeah, so alpha 1 and alpha 2, they both bind epinephrine and norepinephrine. Alpha 1 is responsible for stimulating the adrenergic system. Alpha 2 is responsible for inhibiting the adrenergic system. Why would we need both? Too many things. Okay, so say that again. We'll go one at a time. Yeah. So if the receptor, good. So the concept of receptors being at different concentrations is important because in some, and also the response with a tissue type. So in some tissues, you have more alpha 1 than alpha 2. Those tissues might be more susceptible to an activation. Also, so that's true for other tissues too. So good. What were you going to say? Yeah. So general negative feedback. Right. So general negative feedback to because in general, alpha 2 is going to be less than, or the receptor concentration and the response level of alpha 2 is going to be less than alpha 1. But if you stimulate it enough, alpha 2 will still be a lot to where it can actually turn off alpha 1 and just kind of make an equilibrium. And wait, what were you going to say? Oh, okay. Okay, cool. Good ideas. That's exactly right. Negative feedback, different tissue types, receptor concentrations are different throughout the body. So exactly. Okay, so when do we use epinephrine, right? EpiPen, who's ever heard of that, right? If you, like in elementary school, there was always someone who was allergic to peanuts, right? Is anyone here allergic to peanuts? Yeah, my sister's allergic to different types of nuts. Not like deadly, not airborne, but she never had an EpiPen. But people have EpiPens, and what EpiPen is it's epinephrine. And it can treat bronchospasms, which happen if someone has an allergic reaction. Their esophagus can inflame, right, and swell, and then you can't get any air in, right? So an EpiPen, what it does is it increases the blood flow everywhere, and it can decrease, it can increase respiratory rate, decrease the swelling, and it can increase your airflow. Anaphylactic shock, same thing. Cardiac arrest, local anesthetics, kind of. Well, yeah, if you, yeah. So yeah, it can be used for a lot of to kind of stimulate the body when we have different conditions where we can't get any air in, or we have a heart attack or cardiac arrest. So it could be used for a lot of those different cases. And if it's B1 or B2, it just means beta 1 or beta 2, which receptor is targeting specifically. Anything broncho-related, beta 2, anything anaphylactic shock or cardiac arrest, anything heart-related, beta 1. Yes. Yeah. So no. But you're right that it does, but we don't need to know about that too far. Yeah. Can we use the inverse agonist? Yes. For what? But do the opposite. There wouldn't be inverse agonists for these because so the inverse agonist in general is a difficult concept because it's somewhat subjective, because you can say that if you don't increase the heart rate, you're decreasing the heart rate. In that sense, yeah, it's kind of you can have an inhibitor that's an inverse agonist. So that's possible. Yeah, we'll go with that. So yes, yeah, we'll go with that. For a lot of these functions, like heart rate, for example, you can have inverse agonists. One lowers versus raises. So yeah. Okay. Yes. Yes. Opposite effect. Yes. So there's also, I mean, in cardiac arrest, one of the main reasons might be a blood clot. So if you have any kind of now going into cardiac diseases, if you have any kind of ischemic heart disease, right, which means you have some kind of a cholesterol block, some kind of lipid block in your arteries, this will not help relieve that. That's the main reason. But it will, in general, attempt to increase your cardiac output. But if you have a blood clot and some of your heart has a decreased oxygen, increasing the heart rate is not going to do much because you have no oxygen there in the first place, and you have a clot there. So for that case, you would need to use different types of maybe like nitroglycerin. Yes, that's what I'm saying. Yeah. Use nitroglycerin or different kinds of nitrates, different kinds of nitrates. So that would help too. Yeah, there's a whole class, different classes of those drugs. So I can't check. I don't think about it right now. All right. So anyway, do we have a chapter on cardiac drugs? I think we do. But if we do, then we'll talk about that then. Okay. Cool. All right. Any other questions? Yeah, sure. Sure.
So we're not, you wouldn't refer to alpha 1, alpha 2 as inverse agonists of each other. They're receptors, right? You wouldn't talk about receptors in that.
Right. No. So that's only for a drug. Right. But you have, in this case, you have two receptors that have the opposite effect.
And bind the same thing.
They bind the same thing. Right. But they're not inverse agonists of each other. They are, one's a negative feedback of the other. I'll say that. Yes.
So we only refer.
For a drug that binds alpha 2, so if you have a drug that just binds alpha 2, it would be a direct antagonist to the adrenergic system. So it would be a direct sympatholytic. Right. Even though it would be some kind of epinephrine mimetic, it would be inhibiting the pathway. But I don't think they have one that binds to alpha-2 but does not bind to alpha-1. But that would be a really cool discovery. Well, theoretically, they're different proteins. Yeah. Good questions all around. All right, so clonidine. So clonidine is a drug. It's an alpha-2 agonist that's used to treat hypertension. So an alpha is, to your point, really. So it's mainly alpha-2. So an alpha-2 agonist is not the best way to treat hypertension. This is probably the worst way to treat hypertension because we want to use beta inhibitors to do that. That's beta blockers, right? Beta blockers or ACE inhibitors. Those are the best ways. Now, for an alpha-2 agonist, it is, even though it's an agonist, it is considered a direct sympatholytic, even though it's an agonist, because it's alpha-2, because it's activating an inhibitory adrenergic pathway. So clonidine, essentially on the presynaptic alpha-2 receptors, produces the inhibition of sympathetic vasomotor centers and decreasing the sympathetic output. So we're decreasing all the sympathetic pathways with the stimulation of alpha-2. So then a follow-up question, which I kind of just answered: what other adrenergic receptors can be modulated to treat hypertension? So you have beta-1, definitely. That's what beta blockers attack. Even alpha-1 inhibitor would work. Beta-2, not really, but alpha-1 and beta-1 would be good targets. Yes, alpha-1 antagonist. We want to decrease that pathway. Yes. Yeah, I know it can be confusing. Okay, so another one we have asthma drugs. So asthma, you have kind of a asphyxiation kind of thing. You have a closing of the broncho. Yeah. So you have a salbutamol, terbutaline. Yeah, there's a bunch of other ones too. Do I have a chart up here? Oh, I have the chart on the next slide. So basically, I'm sorry, say albuterol. That's what I was thinking of. Yeah. So, salbutamol, short-acting beta-2 agonist for the management of acute asthma symptoms. So you have an asthma attack, right? You take that, you inhale that in the inhaler. That's in the inhaler. What it does immediately, it acts for a short amount of time, maybe like 20 minutes or so, and it increases your respiratory rate, letting you get enough air in, and kind of it's good for your asthma. It's not instant relief. It's not like you take it, it's like, oh, I actually feel better. It's like you're still breathing a lot. You're still but over time, you just after a couple minutes, you start to feel better. Yes. So the corticosteroids. Yeah. So corticosteroids also work for that. Okay. So corticosteroids, their main objective is to decrease the inflammation. And if you have an injection of that, it would work, not immediately, but it would work. So yeah, so that wouldn't be used for asthma or anything.
COVID was like kind of like near COVID before the pandemic. We started the pandemic and got really sick. The doctor gave me like, I didn't want to take prednisone kind of bad. For the first time, then it gave me like one steroid inhaler. Yes.
Right. So there's different ones. Like prednisone wouldn't be the one, but it would be a different corticosteroid in an inhaling form. Right. That would work. That could work immediately. So yeah, that's definitely true. Cool. Or not cool. They had to do that, but the well, I mean, it just decreases prednisone. It just decreases inflammation all around the body at the same. But it takes maybe like 6 to 12 hours to notice a difference. So, yeah. So anyway, so here's a chart on the right going over different beta-2 bronchodilators, so beta-2 agonists. And you have epinephrine, of course, which is very short acting. You have terbutaline, albuterol, formoterol, and then we have salmeterol. Now, for an asthma attack, epinephrine works, right? If you inject it, but I mean, you don't have epinephrine as an inhaler. So that's why people that have a allergic reaction, who also have bronchoconstriction, epinephrine works great because it's intravenous, it's immediate. For something that's an inhaler, terbutaline, or sorry, really albuterol and salmeterol can be used as an inhaler. So, well, really albuterol. That's the shortest acting. And then for formoterol, really it's albuterol standard for inhalers. Yeah, so I'll explain. The red is the onset of bronchodilation, meaning how soon after do you start to feel those positive effects, right? So the idea for asthma, for an inhaler, you want the shortest time. So formoterol would work, but it also has a long duration. So that's what the blue is. Blue is a duration of bronchodilation. So you'll feel the effects is a long half-life, large half-life, which may not be good depending on how frequently you have asthma attacks. And it just, you want to have a quick on, quick off kind of drug. So albuterol is perfect for that. And salmeterol, you would use that it would be better suited for like sleep apnea, where you have trouble breathing the whole night, so you want something that's longer acting. That's where formoterol would work also. Better for sleep apnea. I would give you a chart, yes. Yeah, so that's a good question. That's a fair game question for an exam, but I would give you a chart with the data on it, and then based on addition, which one of these drugs would be better. So that'd be good. That'd be good. Yeah, for albuterol. Yeah, and yeah, and terbutaline, that was the one for uterine relaxant during labor. So we would not use that for asthma attack. That wouldn't work. And I don't think you can take it orally anyway. I think it's an injection. But yeah. So anyway, okay, cool. All right, so indirect acting adrenergic agonists. That's a mouthful. All right, so we break that down. Indirect, meaning it will not bind directly to the adrenergic receptor, not to alpha, not to beta. And it's an agonist, meaning it will decrease the reuptake. So it will keep the adrenaline, keep epinephrine and norepinephrine in the synaptic cleft for a longer period of time. Or it will somehow another way decrease its uptake. So causes the more release, inhibition of reuptake, or inhibition of degradation. So that means the COMT or the monoamine oxidase. It will inhibit those metabolic enzymes. That's what I mean by indirect adrenergic agonists. So amphetamine, methamphetamine, cocaine does that. So here's kind of what we saw before. We also have this receptor or channel. So VMAT2 is a channel protein that allows dopamine to enter the synaptic vesicle to become norepinephrine. If you decrease that, if you inhibit that, you have an increased amount of dopamine, which gets released into the cytoplasm and gets released. More dopamine means less epinephrine, but dopamine is good, makes you feel better. So people who are on methamphetamine, right, you feel that high is from the increased amount of dopamine, from the blocking of the VMAT2, the inhibition of VMAT2. And then something else that's weird is that Also amphetamine and methamphetamine are similar, but amphetamine, it reverses the dopamine reuptake. So not only, this is a good example of an inverse agonist. So instead of blocking dopamine reuptake, it reverses it. The channel goes the opposite way. So all the dopamine that is now in the cell, that was an increased amount from the blocking of BMAC2, can now enter the blood. And can now enter everywhere else in the body. So meaning you have a higher amount of dopamine, right? So that's how amphetamine works. Then you have cocaine, which is not shown here, but our dopamine reuptake process. It wouldn't be it wouldn't reverse the process. It would just inhibit it, allowing dopamine, and do the same thing with serotonin, allowing it to the effective concentrations in the synaptic cleft to increase. So here's some more information on that. So consequently, norepinephrine accumulates in the synaptic space, resulting in the enhanced sympathetic activity and potentiation of the action of epinephrine and norepinephrine. So the fact that, and also coupling that with dopamine reuptake inhibition, you have someone who is feeling a high and is also stimulated, highly stimulated and hyperactive from the accumulation of the epinephrine and norepinephrine. So it's kind of this dual mechanism. Yeah, so it blocks the sodium chlorine channel dependent norepinephrine transporter. So the norepinephrine transporter, the protein, is a channel. Okay. Right. So it's based on the sodium. It's based on sodium transport. Yeah. Yeah. So it blocks the transport protein. Okay. Cool. Any questions on that? Adrenergic antagonist now. So if we have something that's also called a sympatholytic, right? So we could have either direct or indirect sympatholytic, something that is anti-sympathetic nervous system. So these drugs can be reversible or irreversible, and they attach to the adrenergic receptors. Another term for them is the adrenoreceptor, and they prevent catecholamine activation. So this can be, we're talking about direct or indirect. Usually they're direct. You can make an alpha-1 or alpha-2 or beta-1 or beta-2 antagonist. There's a competitive inhibitor. That would be an antagonist. You can also, going downstream, you can create an activator for the COMT, or the monoamine oxidase enzyme, that can increase the reuptake and the metabolism of these of norepinephrine and epinephrine. So yeah, that's that. So here's some examples. You have, these are alpha adrenergic blocking agents. So phenoxybenzamine is used to treat hypertension. Phentolamine, not fentanyl, phentolamine, to treat hypertension during surgery. And then you have some other ones also that are used for similar purposes. So yeah, they kind of, it's exactly what you expect in terms of the physiological outcome, right? We're decreasing the sympathetic activity. All right, so then one of the most common hypertensive drugs, you have propranolol, which is a non-selective beta receptor antagonist. So beta receptor, since it's a hypertensive drug, which beta receptor you think it's going to bind to the most? Yes, beta 1, because it has to do with the heart. But it's also non-selective. So in the clinical trials, or in the really older trials, they found that it binds to beta 1 better, but it also has non-specific effects on beta 2, which is why they call it a non-selective beta antagonist. So it diminishes the cardiac output, so it decreases your blood pressure and also your heart rate. It also has vasoconstriction. It also has bronchoconstriction, which is a product of blocking the beta-2 receptor, which is not good because if you have a patient with asthma or with COPD or some kind of other or emphysema, right? They should not take propranolol if they have hypertension because it'll make their breathing a lot more difficult. So these are kind of things that doctors think of when they're prescribing drugs, knowing exactly. This is why it's important. This is why receptor theory is important, because if you have a drug that binds multiple receptors and one of those receptors or blocking one of those receptors interferes with another ailment, you can be in a lot of murky water with that. So yeah. More about propranolol. Not really too important. So there's different therapeutic usages. So myocardial infarction, which basically means like you have a heart attack. So which means like your blood flow to your heart is compromised. So this would be using propranolol will constrict your peripheral blood vessels, but it also will decrease your heart rate enough to where it will decrease it will decrease your output so that the effects of the myocardial infarction are not substantial. So it'll just lowering everything. It's not a cure. It's just it's one treatment. Yeah. So also it reduces the incidence of sudden arrhythmic, meaning arrhythmia, death after a myocardial infarction. So it's kind of like a preventative drug used in this case. If someone has a heart attack, it'll just kind of dampen the activity of the heart until they get more control of it with other things. Maybe the person needs different types of PCSK9 inhibitors, something to decrease cholesterol or whatever they need. So also migraine, it can be used for hypothyroidism. There's also some adverse effects like bronchoconstriction, like we mentioned, because it binds to beta-2. Arrhythmias, because once you go from a drug that decreases your heart rate and you get taken off of it, what's your heart rate going to do? Once those receptors now bind, it goes from binding nothing or binding an inhibitor to binding norepinephrine. The heart rate's going to increase a lot. So you need to wean off of these kind of drugs because otherwise your heart rate will increase too quickly. You might be subject to arrhythmia.